Welcome to Biotechonomics TV. Today we're going to talk about RNA. Now there's been a lot of excitement about RNA, especially with the recent FDA approvals of the Pfizer and Moderna COVID vaccines. However, if you look at history, RNA for a long time took a backseat role compared to DNA. So I just want to go through the history of this very quickly, just to put this in some context. In 1953, Watson and Crick discovered the double helix structure of DNA. However, they don't know how that DNA translates its messages into protein. That is discovered 8 years later in 1961 when messenger RNA is discovered, and it's understood now how DNA's instructions turn into synthesized proteins. Now, as we get into the 70s and the 80s, there's a lot of enthusiasm and excitement about recombinant DNA, especially with the work that Genentech did, and really when they came out with insulin. And then into the 90s and early 2000s with the Human Genome Project, again, more and more interest in DNA. RNA is sitting in the backseat. You couldn't really get grants for doing RNA research. Everything was centered on DNA. This was the pre-CRISPR days, so it was still gene therapy, but the emphasis on genetics research was primarily on DNA. So how did that change? How did we get from that position, really 20 years or so ago, to where we are today, where messenger RNA is probably the hottest topic right now in biotech? So the first place we have to start is in 1986. And like many things in the world and in science, it was an accident. There was a man named Rich Jurgensen. Rich was an employee at Advanced Genetic Sciences, and he had some purple petunias, and his company wanted to make them even more purple. I think they figured they could sell more with a darker, thicker shade of purple. So he added additional genes to the flowers, expecting that he was going to get more purple, dark purple, deep purple. And what happened? Instead of getting more pigment, he got no pigment. They came out white. Now, this puzzled a lot of people, those at least who read the research, and it really didn't make a lot of sense as to why this would happen. There started to be a theory that developed into the 90s that, well, you have sense RNA, which is the same as the RNA instructions that your cell produces, and you have antisense, which is the complementary sequence, and that somehow something might have been going on with that. So just about 10 years later, Andrew Pryor and Craig Mello, two academic researchers, started to look at the work that Jurgensen had done and believed that it had something to do with using sense RNA that was either turning off the gene, or possibly it was the antisense RNA. And their control in the experiment was double-stranded RNA. Now, typically we think of RNA as single-stranded, and it usually is, but it can be double-stranded, and often when it is double-stranded, the cells will, whether this is in a plant or a person, will see it as a virus and slice and dice it. And one of the accidents that happened in their lab was that the sense and antisense RNA contaminated each other. They ended up producing double-stranded RNA, and in fact, this ended up silencing a gene that they were looking at with worms. So all of this starts by accident. Now, after this, we start to get a lot of interest in RNA interference, right? And ultimately, the work that Pryor and Mello did led to the founding of Alnylam, which got their first venture round done in 2002. Alnylam then got their first FDA-approved drug just a few years ago, 2018, and this year is going to do roughly $500 million of sales. It also gave them the 2006 Nobel Prize. Now, at the same time, RNA therapies were sort of stuck. Right? There was a lot of research that had gone on in not how do you suppress RNA, the expression of genes using RNA, but how do you express different kinds of genes using RNA. So at the University of Pennsylvania, there was a remarkably persistent woman, a Hungarian immigrant named Katalin Kiriko. Can't really pronounce that right, but we'll just go with that. Anyway, she had been demoted at Penn because she was researching RNA. And I said earlier, if you're researching RNA in the 90s, you're not getting grant money. And Penn said, hey, you're not bringing in the dough. And they demoted her, but she was persistent. She kept going. This again being the late 90s, people were still using copy machines, and she found herself sharing a copy machine and fighting over its access with someone named Drew Weissman, who had just completed his MD, PhD at Boston University, and ultimately the two of them started working together on research for RNA. Not for RNA interference, but using messenger RNA as a technique to produce proteins you want as opposed to silencing ones that you don't. And their big breakthrough came in the mid-2000s. Really was Weissman who discovered that if you just change one of the nucleosides in the RNA, you could eliminate the major inflammatory reactions it was causing. And at that point, you could barely even get it into a human trial, never mind some of the struggles it was having with animal trials. And so a major breakthrough in the use of RNA to treat diseases. At that point, obviously thinking cancer, thinking other things, and obviously today it's being used as a COVID vaccine. But this major event occurs. And what happens? What is the great reaction? Nothing. Because they made a great advancement in RNA, which at the time, even 15 years ago, was sort of like, making a great advancement in mainframe computers. No one really cared that much, except a gentleman by the name of Derek Rossi. Derek Rossi read their research a few years later and used that as the basis for founding Moderna in 2010. And then at the same time, Biotechonomics was formed, the company that makes the other vaccine and partnered with Pfizer, through a license of the technology from Penn, where Weissman and Carrico were working. And that is how we got Biotechonomics. And Carrico today is actually has a title of a senior VP there. So the work was initially ignored. It took a few years. It took a company that really, was involved, basically took her technology and drew Weissman's technology. And that's how we got these RNA companies. But I think what's amazing about this is RNA now gets all kinds of venture funding. You read about new RNA companies every day. But it had to work really hard to get to the level it is now and really prove itself out, I think, with these COVID vaccines. Because again, think about it. Think about all the news you hear about gene therapy and CRISPR. There's still, I think, this inherent bias towards DNA therapies. And it's not that one's better than the other. It's just that RNA's often, for a long time anyways, had this backseat position. But that's clearly changing now. But I think this is a fascinating history. And I think it's a great topic to do some more research on if you are interested. And I will do some more videos down the line on this as well. Well, I hope this is useful. Thank you for watching. Please subscribe, and we'll see you next time.